Good evening one and all. I welcome you all to Ganesh IAS Academy. In today's session, we'll be seeing science and technology based current affairs. Let us get into the news articles one by one. The first news for the day is lab grown human embryo model. Okay, why is this particular thing is in news and what is the importance of this human embryo model? And all those details we have to understand, who has developed it and what is its significance. Those details we must have to know. First thing is, recently scientists have achieved a remarkable feat by creating a lab grown human embryo model using stem cells and chemicals. So they have used chemicals and stem cells to produce these lab grown human embryo model, and they have not used, that is without using a egg or sperm. So we need to understand how human embryo is generally made, and then we can compare it with this human embryo model that they have synthesized using stem cells and chemicals. And this is shedding light on early embryo development. Early embryo development is an area which requires a lot of research and development, because that is the phase where a lot of things happen, in the formation of a child. Okay, so a lot of research and development is required in that particular area. Okay, and researchers from Israel, okay, who have developed it? Researchers from Israel have created a model of 14-day human embryo. And what is the significance of this 14-day? That also we need to understand. And this mix of stem cells and chemicals, okay, here we saw, right, they have synthesized it using a mixture of stem cell and chemical, and that mixture is the crucial starting point for creating an embryo like structure. Okay, so all these points are important. Let us get into the details of each and everything. What is human embryo, what are stem cells, and what is the significance of this particular model. Okay, first thing, human embryo. Generally, how a human embryo is made, and what is human embryo? First of all, human embryo is a developing human being, yes, and when we call it as a human embryo, from the moment of fertilization until the end of 8th week of gestation. So such definitions are important. So it is embryo from the moment of fertilization until the end of the 8th week of gestation. Okay, next, human embryo has 3 main stages. What are they? First is the pre-implantation stage, next comes implantation stage, and then third, the organogenesis stage, where the organs develop in human embryo. And then human embryo is composed of different types of cells. This is important. It is composed of different types of cells that differentiate into various tissues and organs. And human embryo is normally created by fertilization of a human egg, that is oocyte, by human sperm. This is very important. Okay, so generally human embryo is created by fertilization of human egg with human sperm, and this is done in the female reproductive tract, or it can be done in the laboratory also. When it is done in the laboratory, then it would be called as in vitro fertilization. Okay, so we'll understand what in vitro fertilization also in this session very briefly. Okay, so this is human embryo in general, that is which is created by fertilization of egg and sperm. Okay, but this model that they have created is from the stem cells and other chemicals. So we need to understand what stem cell is. Stem cell is a cell which is having a unique ability to develop into specialized cells, specialized types of cells in the body. Okay, so these are stem cells, and in the future, how stem cells are going to be used? They will be used to replace cells and tissues that have been damaged or lost due to disease. So if there is going to be any tissue damage or organ damage, then these stem cells can be used to generate tissues and also organs as a whole. Okay, such research is going on at present. Next, these stem cells have 2 unique properties which enable them to do this particular thing, that is they can be developed into specialized cells, right? So what are the unique properties that they have? First thing is they can divide over and over again to produce new cells. So they have this capability to divide over and over again. Okay. Next is, as they divide, they can change into other types of cells that make up the body. This is also important. This point is here linked directly. Okay. That is, they can develop into specialized cells. They can change into other types of cells that make up the body. That is, either other tissues or other organs. So this is stem cell in general. Next, we need to understand about this model, embryo model and early fetal development. Models, these embryo models help uncover errors in DNA duplication, deoxyribonucleic acid duplication and chromosome distribution. So they uncover the errors in DNA duplication. Why DNA duplication is important? That is because researchers have found that DNA duplication abnormalities occur early in the process and it affects cell division. Okay, so DNA duplication abnormalities can be avoided. Those errors can be avoided when we get into the research of these models. Okay, when we understand these models very well. Next is, these models also enable to study the gene function and their roles in fetal development. This is also important. Gene function can also be studied using these embryo models. Okay. Next thing is the embryo models and how is it used in the research. Okay. Studying early embryo development is ethically challenging. Okay. Generally, studying early embryo development is ethically challenging because once implantation is done, then those embryo cannot be taken out of the uterus. Okay, that is ethically wrong. Okay, so once implantation is done, taking out an embryo for research purpose is ethically wrong because it will be considered as an individual, it will be considered as a human being. So it is ethically wrong. Thus, early embryo development is ethically challenging because we cannot do this. In those areas, these embryo models which are synthesized in the lab can be used. Next is, research during these initial stages is vital. Which initial stage we are talking here? The early fetal development stage. Okay, the embryo development stage. It is very important because most miscarriages and birth defects happen during this period. So when we have a great understanding of this initial stage, then we can avoid these miscarriages and birth defects in future. So for that we need embryo. Okay. Next, understanding normal embryo development and genetic factors can improve in vitro fertilization. Obviously, in vitro fertilization is growing these days like anything. Okay. So like in every important streets of a city, you can find an hospital for in vitro fertilization. Okay, IVF. So it is growing day by day. So for understanding normal embryo development and genetic factors, it can improve this in vitro fertilization and it helps researchers grasp genetic, epigenetic and environmental influences on embryonic development. So all these can be achieved using these embryo models. Okay, and it is ethically challenging because embryo cannot be taken out of the uterus once it has been implanted. Okay, this point has to be kept in mind. Next is, it is not used for pregnancy. These models, these embryo models are not used for pregnancy. This is again important. So these models are intended solely for studying early fetal development. Only for study purpose it is there, and then they are generally destroyed after 14 days and implantation is not permitted. Okay. And what is the significance of this 14 days that we talked earlier? The 14-day limit was proposed in 1979 in UK, and this 14 days is equivalent to when the natural embryo implantation finishes. Okay, so this 14 days is equivalent to when the natural embryo implantation finishes, when it occurs naturally inside the female reproductive tract. Then it takes 14 days for implantation to get finished. Okay. Next, it marks the point when cells begin forming an individual. So far it was a clump of cells, but now it will form an individual after 14 days, and breaking off into a twin is not possible after this 14 days. So before this 14 days itself, if at all a twin is going to get formed, then it has to divide. If not, it will be as a single individual only. Okay, so that is the significance of this 14-day. And it is not used for pregnancy, which has to be kept in mind. Next, we have a concept called primitive streak. So what is primitive streak? Ethical considerations change as embryo transition from cell clusters to individual. Like we saw earlier, when it is as a cell cluster, then they have a different ethical consideration, and when it is an individual, that is after 14 days, then ethical considerations are different. How? The ethical considerations become different when it is a clump of cells, and when it becomes an individual, often related to what is referred to as primitive streak. This is called as primitive streak. That is the transition that is happening from clump of cells to an individual. That is called as primitive streak. So primitive streak is a linear structure that appears in the embryo that marks its transition from having a radial symmetry. Okay, so eggs will be having, or those cells will be having radial symmetry. This is radial symmetry. Okay, but it is a transition from having radial symmetry to bilateral symmetry, like in humans or any other insect. Consider this to be an insect, then it can have only bilateral symmetry. Okay, these are the legs, like that. Okay, so it is having bilateral symmetry, like our bodies marked by left and right hands and legs. Okay, if you see in the pictures, you will understand it better. See, this is radial symmetry and this is bilateral symmetry, and this transition from radial to bilateral symmetry is because of this primitive streak formation. Okay, that linear structure formation. Okay, that is called as primitive streak. A question based on primitive streak may be asked in your prelims examination. Okay, yes. So yeah, so with this we have come to end of this particular topic. Okay, the topic that we are going to discuss now is World Duchenne Muscular Dystrophy Day. So what is Duchenne Muscular Dystrophy and why a day for that is observed? Those details we need to understand, and when is that day observed, that also we need to know. World Duchenne Muscular Dystrophy Day has been observed, like it is being observed annually on 7th September, and it aims to raise awareness about this particular disease and also to improve the lives of individuals affected by this rare regenerative illness. This is important. So it is a degenerative illness. Okay, so it is a very rare disease which happens in boys, and then it is a degenerative illness. Next, what is the theme for this year's Duchenne Muscular Dystrophy Day? It is Duchenne Breaking Barriers. This is the theme for this year, and the Department of Empowerment of Persons with Disabilities, which is under the Ministry of Social Justice and Empowerment, is actively involved in creating awareness about this particular disease. All these details are important. Which department and under which ministry it is coming, and how is it giving a push to create awareness about this particular disease. All these details are important. Okay, you can use these in your answer writing. Next, DMD is a rare genetic condition. Yes, we already saw it as rare, and it is a genetic condition which weakens muscle progressively. That is why it is muscular dystrophy, and it affects approximately 1 in every 5,000 boys. Okay, such facts are also important, and the condition results from an X chromosome mutation. Yes, these points are important. So it results from mutation of X chromosome, and it leads to difficulties in walking, which is followed by impairment of other motor functions. So the hand movement, leg movement, and other motor functions of humans will be affected when they are affected by this particular dystrophy. Next, ultimately DMD affects vital functions like breathing and also heart function. How heart function is also affected? That is because heart is also a muscle. Okay, heart as such is also a muscle. Next, currently there is no cure for DMD. Okay, so there is no cure for Duchenne muscular dystrophy, and the treatment aims to control symptoms to improve quality of life. Okay, so all these points are important with respect to this particular disease, that is Duchenne muscular dystrophy. Okay. The topic that we are going to discuss now is need for innovative mosquito control approaches. Okay, so why is there a need for innovative mosquito control approaches, and what is that innovation that has been made recently? Those details we need to understand. Let us get into the details. Need for innovative mosquito control approaches. Rapid urbanization of the world's population, especially in developing countries like India, has led to annual surges in mosquito-borne diseases. Okay, and one of the emerging innovative methods for mosquito control is genetic engineering. So we are going to apply this method of genetic engineering, which involves altering the traits and the behavior of mosquitoes. So this is how they are going to control mosquito spread or mosquito proliferation. Next, what are mosquitoes? They are small flying insects which belong to a family called Culicidae. They belong to a family called Culicidae, and they are known for their distinctive buzzing sound, which they are famous for, and they are notorious for their role in transmitting diseases to humans and also animals, and they transmit deadly diseases such as malaria, dengue, Zika virus, yellow fever, and other viruses and other diseases, which is affecting millions of people every year. Okay, so this is mosquito, and how are we going to control mosquito? What are the new methods? There is a changing landscape of mosquito-borne diseases, and what is the reason for that? One is urbanization, another is climate change. Okay, so rapid urbanization of the global population in developing countries has contributed to annual surges in mosquito diseases like dengue, which we already saw. Next, it is being additionally augmented by climate change. How? Climate change has expanded the territories where the disease are prevalent already. Okay, diseases would have been prevalent already, but now the areas are expanding. That is because of the climate change, and there is an exemplified indigenous cases of dengue in France. Generally, France is known for mosquito-free region. Okay, so it is considered as a mosquito-free region, but then even in France now we are facing indigenous dengue cases, which means it is spreading in France also. That is because of the climate change. I mean, there is an expansion in territory in which these diseases are prevalent. Next, current control measures. What are the control measures that we have? We use mosquito nets, we use insecticides, and we also use symbionts like Wolbachia. So what is this Wolbachia? Wolbachia is actually a bacteria which is present in 60% of the insects, and it includes mosquitoes also. So Wolbachia is a bacteria which is present in 60% of insects, and it includes mosquitoes also. But there is a particular variety of mosquito, that is Aedes aegypti, which causes deadly diseases like dengue, Zika virus, yellow fever, and etc. This Aedes aegypti does not have this bacteria. Okay, this bacteria is not present in this Aedes aegypti. So what they are going to do in this Wolbachia method is they are going to introduce this bacteria into this Aedes aegypti mosquitoes, so that their lifespan will be reduced and their ability to transmit virus will be controlled. Okay, so a question based on this particular method, Wolbachia method, has been asked. Have been asked in the past prelims examination, 2023 prelims examination. Let us face that question also. Before that, we will see this point. Despite the availability of first generation malaria vaccine, insecticide resistance in mosquitoes is a growing concern. Okay, though we are spraying a lot of insecticides for mosquito control, they are becoming resistant to insecticides, which is a growing concern. Thus, there is a need for exploration of newer methods to control mosquitoes. Okay, so that is why we are going for genetic engineering now. Yes, this is the question that I was talking about. Wolbachia method is sometimes talked about with reference to which one of the following? Actually, the answer here is controlling viral disease by spread. Controlling viral disease spread by mosquitoes and using bacteria called Wolbachia bacteria. Okay, so yes, so when it comes to science topic, you need to give importance even to such small important details. Okay, next is genetic engineering for mosquito control, genome sequencing. Okay, actually genome sequencing is very prevalent these days. Everywhere genome sequencing is being done by researchers. So recent advancement in next generation genome sequencing techniques have enabled researchers to obtain whole genome sequence for various mosquito species. So there are whole genome sequence for whole mosquito species available now, and using these we are going to control it. How? This point is important. Research institutions like University of California and institutes in Bengaluru in India have contributed to high quality reference genome for a particular variety of mosquito, that is Anopheles stephensi, which is a major malaria vector. Okay, so such details are also important, and this point is linked here, that is the availability of mosquito genome sequence and our capacity, the researchers' capacity to genetically manipulate them offer unprecedented opportunities for mosquito control. So we have genome sequences and we have capacity to manipulate them. Okay, we are going, we have this capability to manipulate it genetically. So that is how they are going to control this mosquito. And what is the method that they are going to use? The method is gene drive technology. Okay. So what is this gene drive technology? It was developed by Austin Burt in the year 2003, and it aims to manage mosquito populations. How? By changing how they inherit certain genes. Okay, this is important, and it breaks the usual genetic rules explained by Mendel. Next is, this technology uses special protein to edit mosquito DNA. Okay, so it will be using a special protein to edit, which means they will cut a particular DNA, a particular section of mosquito's DNA, and when these protein cut the mosquito's DNA, the cell fixes it by adding a specific genetic sequence. Okay, this is important. So after the cut is made, say for example this is the DNA, and if a cut is made in this part and this part is removed, that part is actually fixed using cells by adding a specific genetic sequence. The cells will add another genetic sequence, okay, a newer sequence, but this newer sequence will affect mosquitoes' ability to have offsprings, and it will also stop malaria parasite from multiplying inside them. All these points are important, and researchers at Imperial College London genetically enhanced a gene in mosquitoes to secrete antimicrobial substances which will disrupt Plasmodium parasite. Okay, this Plasmodium parasite is the one which is the reason for malaria spread and then reducing mosquito lifespan. Okay, so this is how gene drive technology is being used to control mosquitoes. Next we have a mosquito called OX5034 mosquito. What is this OX5034 mosquito? US Environmental Protection Agency has authorized the release of genetically modified mosquito, that is OX5035, in Florida and Texas in the year 2020, and this mosquito is developed with a gene sensitive. It is developed with a gene which is sensitive to an antibiotic called tetracycline. So if this antibiotic tetracycline is given to a person who is affected by this mosquito's virus, then it becomes sensitive to it. Okay, then it carries a self-limiting gene. What is self-limiting gene? That is, it prevents female offspring from surviving, and it will lead to reduction in mosquito population. So this is how this mosquito is going to help in reduction in mosquito population, okay, or mosquito spread. So these points are important, And how is this genetic engineering going to benefit mosquito control? This is also important. Okay, so targeted mosquito control. What? Targeted mosquito control. This targeted approach reduces the need for broad spectrum insecticides, okay, and minimizes harm to non-target species. This is important, okay. So there will be targeted mosquito control so that there won't be harm to any other species. Next is there will be reduced environmental impact when compared to traditional insecticides, okay, because these traditional insecticides will cause more harm to other insects also, and it will also cause a greater impact on environment, but there will be reduced environmental impact when we go for genetic engineering method, and it does not involve chemical pollution in the ecosystem, whereas this traditional insecticide causes chemical pollution in the environment. Next, sustainability. How sustainability? Once released, genetically modified mosquitoes, the mosquitoes that we are talking about now, it can continue to pass on their modified genes to from one generation to another generation. Okay, so this is how the mosquito, the change or the modification is transferred from one generation to another generation, providing a sustainable and self-perpetuating method of mosquito control without the need for frequent reapplication. This is important. So there won't be any need for frequent reapplication. Okay? Yes. Next, what are the risks and the concerns of genetic engineering for mosquito control? The main risk here is ecological imbalance. So, okay, so altered mosquito population may disrupt food chain or create ecological imbalance. So everywhere, if you see, even when it comes to ethical concerns, then it is again focusing on the question of ecological responsibility. Okay, and then the risk of invasion. Again, ecological disruption in the regions outside their natural range might happen. Okay, when we go for genetic engineering based control of mosquitoes. Okay, so these points, this ecological concerns and ethical concerns, and there are risk of invasion. So such points are important. Yes. So with this we have come to end of this particular topic, that is genetically modified methods for controlling mosquito. Okay. The topic that we are going to discuss now is Nipah virus. What is Nipah virus? Why is it in the news after nearly 2 years? Let us understand. Okay. Nipah virus has made a resurgence in Kerala in India with 2 fatalities. That is the reason why it is in news, and this marks the first Nipah virus outbreak in India since 2021. Okay, so the last case was in 2021 when a case was reported in Kozhikode of Kerala during COVID-19 pandemic, and after that only now we are facing this Nipah virus outbreak, and it is a zoonotic disease, a zoonotic virus, which means it is transmitted from animals to humans. That is zoonotic disease, and this Nipah virus is a RNA virus. That is important, and it belongs to the family called Paramyxoviridae, and then genus Nipah virus genus, which is closely related to Hendra virus. Okay, so this Nipah virus or the Nipah virus is closely related to Hendra virus. Then what is Hendra virus? Hendra virus infection is a rare and emerging zoonosis, that is a zoonotic disease that causes severe and often fatal disease when it is affected by, when it affects horse and humans. Okay, so this is Hendra virus. So Nipah virus and Hendra virus are closely related. Such statements are important. And when did we see the first outbreak of Nipah virus? The first broke out, it first broke out in Malaysia and Singapore in the year 1998 and 1999, and the disease is named after a village in Malaysia, that is Sungai Nipah. Okay, where it was first detected, and it first appeared in domestic pigs, and it has been found in several other domestic species like cats, dogs, goats, horses and sheep. Okay, so these topics are important. And how is it transmitted? How is this Nipah virus transmitted? This disease spreads through fruit bats or flying foxes, okay, of the genus Pteropus, okay. And these fruit bats are natural reservoirs or hosts of Nipah and Hendra virus, both. And the virus is present in bat urine, bat feces, saliva and also birthing fluids. And what are the symptoms? Human infection presents an encephalitic syndrome. So what is encephalitic syndrome? Encephalon means brain, okay. Encephalon means brain and encephalitis means inflammation in the brain, okay. And it is also marked by fever, headache, drowsiness, disorientation, mental confusion, coma, and it can also cause death in some cases. And how is it prevented? At present there are no vaccines for both humans and animals, and intensive supportive care, ICU care, is given to humans who are infected by this Nipah virus. Okay, so that is how Nipah virus is controlled. Okay, so yes. So with that we have come to end of this topic, Nipah virus. The next topic that we are going to discuss is natural polyphenol to combat Alzheimer's disease. So what are these polyphenols we are talking about, and what is this Alzheimer's disease that we need to understand? Researchers at Jawaharlal Nehru Center for Advanced Scientific Research, which is an autonomous institute of the Department of Science and Technology, they have discovered a naturally occurring plant-based polyphenol. What is this polyphenol? A beneficial plant compound with antioxidant properties, and the example given here is tannic acid. Okay, so the polyphenol like tannic acid, which is found in chestnut and oak twigs, can yield safe and cost-effective strategy for combating Alzheimer's disease. Okay, and what is Alzheimer's disease? It is a progressive neurodegenerative disorder which is characterized by memory and cognitive decline. Okay, so that is Alzheimer's disease, neurodegenerative disorder, which has this characteristics of memory and cognitive decline. Okay, and this Alzheimer's disease is linked to another disease called as ferroptosis. What is this ferroptosis? It is a type of programmed cell death which is dependent on iron, okay, and is linked to development of Alzheimer's disease. So Alzheimer's aligns with the characteristics of this ferroptosis, which includes abnormal iron buildup and reduced activity of a particular antioxidant enzyme called glutathione peroxidase 4, okay, that is GPX4, glutathione peroxidase 4. And how is this tannic acid, the polyphenol that we talked about, is going to help here? This tannic acid activates and enhances this GPX4, offering a comprehensive approach to combat Alzheimer's disease. So you need to understand what Alzheimer's disease is and to which disease is it is closely related to, and how are these polyphenols going to help in combating Alzheimer's disease. Okay, so this is what you need to know from exam point of view. Next is India and WOAH collaborate for pandemic preparedness. So what is this WOAH? World Organization for Animal Health. Why are they collaborating? And that too, why for pandemic preparedness? Let us understand, and we need to understand about this particular organization also. India and WOAH collaborate for pandemic preparedness. Recently, Department of Animal Husbandry and Dairying, in partnership with this World Organization for Animal Health, has organized a multi-sectoral workshop with the topic risk-based management for spillover events in wildlife in India, where at Hyderabad. Okay, And this workshop aimed at enhancing stakeholder knowledge about wildlife origin disease risk analysis, and this discussion emphasizes on the significance of One Health approach, which we have discussed many a times in our session. And what is a One Health approach? Which recognizes interconnectedness of wildlife, domestic animals and human health, and it remains crucial for sustainable future. Now let us understand the importance of this organization, that is World Organization for Animal Health. It is an intergovernmental organization which was founded in the year 1924 to improve animal health worldwide, and it aims to control and prevent the spread of epizootic diseases. Important. And then this WOAH, that is World Organization for Animal Health, has 182 member countries including European Union member states, and this has its headquarters in Paris, France, and there are many regional commissions which are maintained in each region of the world. This is important. Okay, so what are epizootic diseases here? Those are temporary spread or temporary spread in diseases among animals. Okay, so that is called as epizootic diseases, and this organization aims to control and prevent the spread of epizootic diseases. Okay, so with this we have come to end of this particular session. I hope you found the session to be very useful and informative. Thank you. Let us see in the next session.